Good evening, everyone present here with us, and welcome you all to today's webinar. Myself, Dr. Deviprita Dotto, Assistant Professor, Department of Zoology, Gurucharan College, Silchar. Today's national webinar on research opportunity in herpetology with special reference to Barak Valley is organized by Department of Zoology, Gurucharan College in collaboration with internal quality assurance cell of our college. We are really grateful to them for helping us. The whole session will be live on YouTube through StreamYard, and a recorded version of the same will be available for all of you on on our YouTube channel GCC Academia for future use. Now I would request Dr. Sheba Roy, Madam, Head Department of Zoology and convener for this national webinar, to welcome our guest with her words. Please, Madam.
Thank you, Devi Prita. Hello, everyone. Very good afternoon to everyone. I hope I am audible to everyone.
Yes, Madam, you are.
Okay. So, today, on behalf of the Department of Zoology, Gurucharan College, and IQC of our college, I'd like to welcome one and everyone present in this webinar. First of all, I'd like to welcome our principal sir, who actually encouraged us to hold this webinar. Thank you, sir, and you are most welcome to our program. Secondly, I'd like to welcome Dr. Abhijit Das from Wildlife Institute of Dehradun, Wildlife Institute of India, Dehradun. Actually, we know him as a snake man, and he is very popular to us. He is also an alumnus of our college as well as our Department of Zoology. So I welcome you, Abhijit, to our program. Next, I'd like to welcome our... Am I audible, Abhijit?
Yes.
Can you hear? Okay. Next, I welcome
yes, now you're audible.
Okay. Next, I welcome IQC coordinator, Dr. Rajashree Pal, who is not present in today's program due to some, some internet connectivity, internet issues. She is unable to be available in our program, but still, if she gets connectivity, she will come back. Next to Rajashree Madam, I'd like to welcome all our faculty members of Department of Zoology as well as other departments. Faculty, I hope other department faculties from other departments are also present in this program. I don't know. I don't see them. I can't see actually on screen, but still they might be present. They might be hearing us. So I also welcome them. All our faculty members, welcome. I hope Freeman is there, but I don't see Freeman. I can see Freeman, but hope yes.
I'm
okay,
present.
Freeman. Okay. Welcome you, Freeman. Welcome.
Thank you, ma'am.
Okay. Then next, I shall be failing in my duties if I do not welcome my dear students. Especially, I'd like to mention the name of Visho. I could see him. He's on screen. And probably one more student, Aradhana, will be joining us shortly. Isn't it, Visho? I hope she will be joining us. Okay. No problem. I welcome you all. Then I hope all our students are hearing and listening to us. And also, we are most eagerly waiting to listen, to hear from our snake man, that is Dr. Abhijit. He will deliver his lecture. Also, he will present some slides. So thank you. And lastly, I should mention the name of Debashish Sharma, Dr. Debashish Sharma, also the also a few faculty members who are working backstage, like Keshav Luitel, I think, and then Nirupam, who are working on the e-certificates. I also welcome. I don't know whether they are present here. They are listening to our program or not. I don't know, but still, I welcome them. And lastly, whoever present in this program, who are listening our program, and who are eagerly waiting to listen from our speaker, today's speaker, Dr. Abhijit. I welcome you all technicians, all who are present. I welcome. Thank you very much. Thank you. Please, Devprita.
Thank you, madam. So I would now request head of this premier institution, our principal, sir, Dr. Bibhash Dev, to say a few words on this occasion, sir. Please.
Well, first of all, namaskar and good afternoon to all of you.
Good afternoon, sir.
Good afternoon, sir. Yes. I welcome all of you in this national webinar on herpetology. The topic is recent opportunities in herpetology with special reference to Barak Valley. Now, respected HOD Madam, Dr. Shebarai, Dr. Rajashree Pal, Coordinator IQSC, other member of Zoology Department, Dr. Deviprita Dotto, Dr. Nilendu Dhar, Dr. Srimen Boro, Dr. Samrat Sengupta, and most importantly, the center of attraction, that is Dr. Abhijit Das, is with us, and he will deliver his speech on herpetology. This is a very important topic chosen by the HOD and his team for this webinar. Actually, during this pandemic, our all academic exercises has been stopped and only online exercises are on. For that reason, that is, this webinar will be most beneficial to all the students, teachers, research scholars, particularly in this field. I thank Dr. Abhijit Das for responding to our call, and I believe after his deliberation, all will be very much benefited, especially our students, our scholars will be enriched, and they can proceed towards this line. On this line means herpetology, to do work, to do research work, to do publish some papers. Since long back, that is, we are trying a YouTube channel. Ultimately, our IQSC, with the help of the IQSC core committee member and the other committee member, Devashish Sharma, Nirupam Malakar, Arjun Choudhury, all have tried their best, and ultimately we got a channel, YouTube channel, in the name of GCCE Academia. So everybody will be able to see the our activities, our academic activities, our college activities. Now, Zoology Department is very, very much active in this regard. Good number of major research projects they have done. They have published Biovision as their departmental journal, and they are also doing excursion, outreach program, awareness program. So thanks to Dr. Shebarai Madam for doing all these things, and I believe they will carry out all these activities. As A grade college, Gurucharan College is an A grade college accredited by NAC Bangalore. We should increase or enhance these type of activities during this COVID-19 pandemic. And the students, I believe, good number of students, good number of scholars have participated by this time. And please hear from Dr. Abhijit Das so that you can enrich yourself. I don't like to lengthen my speech because it is not my subject, herpetology. I am a botanist and finally I am a microbiologist. Still, I got some amount of interest because it is a biological matter or biological subject. So finally, once again, thanking Dr. Abhijit Das and all the organizers, that is Department of Zoology headed by Dr. Shevara and all other members for organizing such a beautiful, nice and a nice topic. So with this, I would like to conclude. Thank you once again.
Thank you so much.
Very much.
Those were really wonderful words from you. So now let me introduce our today's presenter, Dr. Abhijit Das, Scientist D, Wildlife Institute of India, Dehradun. So before Dr. Das starts his presentation, let me share a few words about him, which I got from Madam. So, Dr. Abhijit Das is an alumnus of GC College, Silchar. He did his graduation with zoology as honors from this institution only and completed masters from Guwahati University. He got his PhD from Utkal University, Odisha, and after that he joined Wildlife Institute of India, Dehradun in 2013 as Faculty of Wildlife Sciences. His research interests include morphological and molecular systematics of herpetofauna, biogeography, and conservation. So without further ado, let me welcome Dr. Abhijit Das. Dr. Das, the stage is all yours, please.
Thank you, madam.
I would like to add one point here. Abhijit did his PhD on snakes.
Okay.
Abhijit did his PhD on snakes, and particularly snakes of Northeast India. And he has interest in studying snakes in Borail, isn't it?
Yes,
Dr. Das?
yes,
Okay.
yes.
Thank you. You please carry on. You are welcome.
Okay, thank you, ma'am. I think I am audible, and
Yes.
I will share my screen. And can you see my screen?
Yes.
Yeah.
Now this one is okay.
Now, if I am going to presentation mode, can you see that?
Yes. This is the
Yes.
slide. This one is the slide.
Okay. So first of all, thank you all. Thank you so much for calling me, and I really appreciate this because I feel this is my greatest opportunity to talk to students from my alma mater, GC College, Silchar, and you know,
Dr. Abhijit, I will just
Yes.
interrupt. Can you please
Yes.
click the high definition hide option? Yes, hide option in your screen. On your screen, there is a hide option. Can you just
Yes.
yeah, that pop-up will go.
You can see my screen still
No, it's not visible.
now.
Yeah, now it's okay. Yes.
So when I click hide, it just goes. Everything goes off.
Okay, then leave it. Okay, you can continue with that one.
Okay. So thank you all, and first of all, I just want to quickly thank all my teachers, Kalpana madam, Mitra de, Baby madam, Sheba madam, our very popular Nilendu sir, Shankar Bhatt sir, and Chaya madam, and of course the D companies, Debuda and Durgada, I think, yeah. So they helped me a lot during my college days, and I can't forget their contribution to my life and my career. So today we are going to talk about, as Principal Sir said, herpetology and something, to deal with reptiles and amphibians. Although I did my PhD in snakes, but I have different priorities in this subject. For example, snake is my first love, but obviously for answering various other questions, I do use other models like frogs and turtles and lizards. So as a whole, I like this subject, and I'm professionally associated with it. What I wanted to say is that you all must have a ecology syllabus, right? And this was exactly when I was a student. This was also in my mind. Like, we read so many topics, say for example, principles of ecology, biotic and abiotic factors, population density dependent factors, community ecology, social behavior, and so many things, right? But we are human being, and we like something which we see in front of our eyes, and which we can prove. That is the something which we like, and that's why there are more engineers, more doctors, more physics students, and chemical engineers rather than ecologists. One of the reason is that what we read in our college and universities, we cannot relate those studies and topics to the research. For example, many of us, we read density dependent factors and population regulation. We write the same thing in our exam, but we never test them, because those principles have come to our ecology book from research in America or research in in Australia. So, and we have nothing to do, so we just read them, write in an exam, and forget it, and then we join microbiology and biotechnology. But if we can able to test those things, so we will definitely love ecology, and we will see that ecology is around us, and this is one subject which we can actually experiment with organisms like frogs and snakes and fishes and insects, right? So these are all just around us. So that's what is my message, that you can, what we read in our syllabus, we can actually do exactly the same thing with these animals. So, and they are just around us, and we will talk about some of them in our Barak Valley. Am I audible so far?
Yes, you are audible.
Yeah. Okay. Thank you. So next slide, and the first slide is actually Northeast India, right? So people living in Northeast India, that means you all are very, very lucky people, because you live in a place which is part of two global biodiversity hotspot. And I like Northeast India. My home is there, but I realized or missed Northeast India more during this COVID pandemic, because for one whole year I'm not able to go to Assam. And then I realized that when I go to Assam, I used to go sneak out of my home, and I used to find 10 species of frogs near my home. Or else I just, in half an hour from my home, I can go to Bareil, and in one night I can see 30 species of frogs and snakes. And in just one night, so many things. So I feel that everyone living in Assam have this privilege of seeing 30 species of reptiles and amphibians in one night, if there is a little bit of effort of just going out of your home. So that's the beauty of Northeast India. It's a hot spot. You have so many species, and they are just your backyard. You need not to go to a large protected area or Kaziranga National Park to see them. They are just there. Now, biogeographically, or say from wildlife perspective, this region is very, very interesting. If you see the Northeast India map, you see the Arunachal Pradesh, which is a part of Himalaya, and you have this large plains of Assam, which is 80-kilometer wide. That is called Assam Plain, where or we also call it as Brahmaputra Valley. And then we have lot of hills towards south of Assam. That means these are called northeastern hills, and they are Meghalaya, Nagaland, Mizoram, Manipur, Tripura, and also there is a very, very specialized hill range called Barai Hill in between, right? So what is the specialty of Northeast is that it is a faunal gateway. That means the Indian fauna went outside, or from outside the fauna came inside in India through Northeast. In Northeast India, we get fauna from China, fauna from Myanmar, and also from Indian region. So it's a mixture of everything. In Northeast India, we have the great elevational gradient. So you have Tripura or Brahmaputra plain, which are 30 to 40 meter in elevation, and you have the highest peak in Arunachal Pradesh, which are up to 5,000-meter elevation gradient. And in some area, if you start walking in the date early morning from 100-meter elevation, by evening you reach 3,000 meter. So you have one single gradient of elevation. So just imagine the opportunity of our research. We can actually do a lot of research in this area with so much of variation in physiography. And then you have Brahmaputra river, which separates fauna of northern side and southern side. We have deep valleys. We have Barak river separating fauna of Brahmaputra valley and Barak valley. So many things are there. It's the stage is all set for researchers to just go and explore. But we were talking about a hill range which is of our interest, that is called Barai Hill Range. So if you see the position of this hill range, Barai Hill Range starts from south of Nagaland, and then it comes to Assam, and then goes all the way and join Meghalaya. So the people who have gone to Shillong or Guwahati through Sonapur, so you must have seen the hill area near Sonapur where a lot of landslide happens. So these are the area where Barai Hill goes and joins with the Meghalaya hills. So it is like a bridge between Nagaland and Meghalaya, in between that. And in this Barai Hill also, in the place where my village is called Borkhola, so that area is like 20 to 30-meter elevation only. And it goes up to 2,500-meter elevation in Nagaland. So this is a really a great elevational gradient where you can actually have this large hypothesis to test. As a PhD student, you can take it up. For example, say as you go from 50-meter to 2,000-meter elevation, is there a species number that are going to increase or going to decrease, or species number going to increase up to mid elevation and then slowly start declining? We call in ecological principle mid domain effect. So these are great things to do in Northeast India. And, these are some of our thrust area in research. However, we are going to talk about something called herpetology, and probably now you are clear that they are uh, reptiles and amphibians. So although amphibians and reptiles are very, very different, but in their lifestyle, but as a group, they are considered into a single branch of science called herpetology. And some of the numbers that you see inside these figures, these are the species number, okay, within this group. So frogs in India, we have some 430 species. We have 3 crocodiles. We have 36 species of turtles, which is the highest in the world, one of the highest in the world. We have a lot of lizards and snakes also, although snakes, some only few of them are venomous. So and obviously for anybody beginners, I also want to say that none of the lizards in India are poisonous also. So what is special about this group of animals? So these are, one of the specialty is that what we call cold blooded animal. Okay, So, but that does not mean that their blood is cold. The cold-blooded animals means the way they maintain their body temperature. So the cold-blooded animals or these reptiles and amphibians are dependent on external light source for generating body heat. So that's why these animals comes out in the morning under sun, and what they do basking. So that's what they maintain their body temperature. Although we may think that these are very primitive animal, but it is, although they are primitive, they have started living before dinosaur in this world, but still they are very very successful in this world. There are more than 8,000 species of frogs are there, so they are very very successful animals, and they are still with us. So what is the why they are so much successful? The reason is that their life is in a very, very low energy demand. That means you don't give any food to any of this, but still they will survive for many months together. For example, they go for hibernation in the winter months. So in winter season, a bird, if does not migrate, they will die. But a frog or a snake, what they will do, they will go into hibernation and they will not eat for maybe 6 months together, but still they will survive. So that's a unique capacity. So for just imagine, if we would not have needed food for 6 months, then we would not have needed any job also. We just sit and just be enjoy life, right? But for reptiles and amphibia, this is possible. They One reason is that their way of lifestyle is very very different, because they are cold blood. They bask in the sun. Their mode of communication is mostly chemicals, but as a mammal and bird, they mostly communicate through voice and color. But in reptiles and amphibians, it is mostly chemical communication. So we get very much difficulty in interpret them or understand them. That's why reptiles and amphibians are very very poorly known. Because it is difficult to find them, because it is difficult to know what reptiles or a lizard or a snake is thinking or behaving, because we can't know. That's why we are difficult to understand it in ecological term. So that's why they remain as the most poorly known group of reptile. Some of the information that are available with us. For example, there are a lot of amphibians that live in the forest. There are a lot of frogs or many other reptiles also. They live in the fast flowing streams. Some of them live in the forest floor. That means the leaves that fallen off on the ground in the forest, they live in that leaf litter. So these are some information available. But when I show you this graph, so you say that in Northeast India, we are talking about Northeast, there are a lot of species that you see the last bar that is not known. So there are almost 50 species of reptiles and amphibians, but we don't even know where they live. So that shows that how much our knowledge in this group is poor. We don't even know where they live at all. Okay. But some of them information that we have, they live in lotic habitat, that means fast flowing habitat. A lot of species in Northeast live in leaf litter, as well as arboreal species. Some are rupicolous, that means they live on rocks. Some are terrestrial, some are lentic, that means they are on shell, ponds and pools, and few of them are burrowing also. So you as a researcher, and that will be a great information to tell to this world, right? Because conserving these animals are really difficult without basic information such as where they live, what they do, what they eat. These are the basic information that we need if we want to save any species. Okay, so that's why it is very important for us to know about them before talking about conservation. Okay, so that's a message for this particular group. And this is true not only for smaller frogs, this is true This is also true for larger species such as tortoises and turtles. For example, this you see this peacock softshell turtle that may grow up to really large size. Some of the softshell turtles can grow up to 50 to 60 kilogram weight, as much as a human weight. You know, some very large tortoises, for example, you know probably the difference between turtle and tortoises, right? Because tortoises are hard shell, they live on the land, and turtles are soft shell or hard shell that lives in the water. So this giant tortoise that is found in our Baral Hill area, we don't even know how many are left in the wild. What is the population? What kind of food they eat? How much they move? And and what kind of threats uh, are there in our area? So we don't even know. And these species are going extinct. They are all endangered or critically endangered because they have a very long generation. They get matured at a very late age, and before getting matured and start breeding, they are being harvested, okay, because they are eaten. So that's why they don't even get chance to breed. So that's why the population is crushed, and they are all in extinction crisis. So even the condition with a large species like a turtle and a tortoise is same as frog and a lizard, which are much smaller in size. So they are called as covert species. Covert species means species that are hardly known. Okay, so and covert species are the most difficult species to conserve in our popular parlance. So we need to know a lot about them to save them. So we need to do research on them, right? So what, say for example, there must be some prerequisites for research that is needed. You need to have aim, okay? You need to have an aim. For example, maybe your aim is to conserve the reptiles and amphibians of Baral Hill. Okay, this is your aim. Aim is such a large vision. But for achieving that aim, you must have some objectives. So what are the objectives? I think if you want to save, conserve the reptiles, then one objective should be you need to know how many species of reptiles are there. Second objective is that what are the habitat they occupy. Third objective is that what are the population sizes of these reptiles. And fourth objective is probably to make aware people that not to kill, not to destroy the habitat of reptiles. These are four objectives you have, right? And then you need to have methods specific to those objectives. For example, for first method, that is inventory, you need to have survey which are designed only for inventory. Second, for population, you need to have some method which are designed only for population estimation. So that you need to do. And one thing you need to remember, that you need not to develop your own method, but you need to follow standard method that are already followed in research. Otherwise you cannot compare your result with others. Then you will go to forest, you will collect data. But unlike any other branch of science, wildlife data collection is a very important aspect, because in forest and in remote area in Baral or any other place, you will hardly get one chance to collect data. And if you misplace that data, if the data goes off or you lost the data, computer crash, hard disk crash, then you lost the data, you lost everything. So you have to collect data properly as well as maintain the data properly. So it is not that you can create another set of data. The tortoise or a snake will not come again to show you off in the forest. It is just one chance. Then you need to know limitation of research, how much funding you have, what is the time frame that is there for your research. Okay, so these are some limitation you need to know. For example, you cannot plan a reptile and amphibious study in winter season because you will not find any. Or you cannot plan a very large, say, herpetofaunal inventory of Baral Reserve Forest in just 6 months of your dissertation. It needs more time, right? So these are certain limitation you need to know, and no research is research unless you publish your finding. And you should always strive to publish your research in good peer reviewed journals. So these are basic prerequisite, but if you maintain this, not only this helps in your research, but it also helps you get research grants. Many of you probably have this, idea of having your own research project. You want to be independent, you want to do what you like to do, just like me. So probably that is the case. I mean, in that way, if you want to develop your research grant, you must have to write it nicely. I remember my days when, after my MSc, when I selected Bareil as my study area, there was hardly any research, any sampling done in this landscape. So it was a challenging for me to uh, get research grants from United Kingdom, Rufford Foundation. But when I came to know that in Boreal area you have a forest type called Kachar tropical evergreen forest. So that particular forest type is unique and only found in the whole world. It is only there in Boreal. So I made a story around Kachar tropical evergreen forest, and I have made these frogs and snakes and lizards as the star of this forest, and I presented it in front of the research board funding bodies, and they accepted it. And they not only accepted once, they have accepted consecutively 3 research grants on Bareil. So that's a really great thing. So you all can do. There are a lot of opportunities, because our area, or Barak Valley, is least explored, one of the most you can say poorly explored area, very less priority, but still, because this is less studied, you have more opportunity to do research. Okay, so to do herpetological research also, you need some very basic field materials, for example. And with this simple field materials, you can do wonders. For example, you probably need a small vernier caliper. It may be a digital caliper also, or a dial caliper. So for measurement, this is for accurate measurement of the this is used for taxonomic research as well as ecological research. Because you will be mostly working in the rain, because herpetology is a study of rain, time of rain. You have to be in the field, so you need to have those notebooks or ink which are waterproof. You will be working mostly in the night, so you need to have a headlamp. Okay, the place where frogs and snake live, they are infested by leeches. So you have to go to forest with proper guard, because in Bareil area, if you go with nice shoes and socks, and you cannot walk half an hour, there will be so many leeches, so you have to be properly equipped. You probably need a camera. SLR with macro lens is best, but you can begin, like me, you can begin with a small camera, digital camera, which you can take picture in the field. Also, if you can buy some equipments, like this is weather station, mini weather station, Castrol meter. So with that you get humidity, temperature, moisture, airflow, every ecological data what in your syllabus. In you go back to your zoology syllabus, you find biotic and abiotic factors. So you can collect all abiotic data with this equipment, right? Then if you have song recorder or voice recorder that is used by reporters and everybody, so you can buy a small call recorder or acoustic recorder. That is great. Oh, okay. If I mean, this is something which you can take a call. This is a spring balance, so it's a field spring balance. So you need, with that, we keep a frog in a polythene bag, and then we take the weight of the frog. So that is for ecological study. So that is called Pesola balance. And if somebody is interested in snake, he or she must have to go to forest with a snake hook. So these are very basic equipments with which you can do wonderful research, and you publish a lot of research paper Okay. They look a lot of things, but together, all together, their cost will be less than a bike. So you can choose to buy all these equipments or buy a bike. Okay. So not that costly. So you, with all these equipments, you go to forest. If you go to forest area in Bareil, the best time to go is February, March, when most of the area is dry. So you, in Bareil, unlike Kaziranga and Manas, you don't have roads, forest roads, so you have to walk along the streams. So March, February, March, the streams are mostly dry. Okay. So these streams are dry and you can walk along this stream. But as a biologist, and if you are working in herpetology, you have to be careful in observation of microhabitats. For example, when you walk on the stream, there will be so many interesting things that for a general public it will miss, but for a biologist or a researcher, you must have to observe that there are various kind of rocks available in that area. For example, the large rocks that are called bedrocks, you cannot move a bedrock. There are boulders, there are small rocks we call as pebbles. There is a pool of water. Cobbles are, say, 30 centimeter big rocks. They are called cobbles. So you can differentiate all this microhabitat. Now, if you go to after April, March, April, May, June, the rain starts in Bareil. So most of the stream will have water. So the area of the stream where water is dancing around, so those areas are called pools. Sorry, refills, and the water that is not moving in our area is called pools. And along the stream, you get so much thick vegetation, and in those vegetation, you see sometimes snakes are hiding or lurking behind those bushes. So you have to be very, very careful walking on those streams. Okay. So, but this is the opportunity, because you just see along the stream, these animals are using those riparian vegetation. Okay. So you have all the stage to set, and just you need to go and explore this area. But if you were a herpetologist, you have to go to forest not on the mostly in the daytime, but also mostly in the night time. So what do you do going in the forest in the night time? So there is a survey method, we call it visual encounter search. There are a lot of survey method, but this is very, very popular survey method. So it's called VES. So what we do in the forest, there are one or two observer. We, with the help of torchlight, we walk from a bottom of a stream to above the stream. And visual encounter search means it's visual and it is time constraint. For example, we just keep the survey for 1 hour period. Why 1 hour? Because in the night time, if you, with all your heart and soul, if you start searching in an area for 1 hour, after 1 hour you get tired, so you start there developing biases in your observation. So 1 hour is good enough time to look for these reptiles and amphibians which are very small in size. So you have to be very, very careful. 1 hour, no disturbance. So that is called time constraint visual encounter search. And you can do as many VES as possible to get better data, right? So we, using all these methods, we have inventoried herpetological inventory of Bareil Wildlife Sanctuary in, and we published the result in 2009. So at that time, I found 45 species of reptiles and some 25 species of amphibians, and the number is still increasing. The number is going much, much higher now using visual encounter search. And in this side, you can see the a data sheet of visual encounter search, where you just need to mention date, time, place, and it is very important to mention the time begin survey, time end survey. So you know 1 hour, and then you have to write the species name, and that is the most tricky part, because you may not know the species, but then you can back up your species with the photograph that you take, photograph number, okay, against the species number. You may, in later part of your research, you may mention snout to vent length, but you never forget writing substrate, location, activity, and time of encounter. So every 1 minute, 2 minute, 3 minute, 5 minute, 10 minute, how many individuals you are finding, what they are doing, where they're sitting. Are they sitting on rock? Are they sitting on leaf? That you have to write. So that makes a visual encounter survey. So just imagine you do 100 visual encounter survey in one area. So just imagine how much data you will have, and you will know exactly this, the preference of the species, whether that species prefers sitting on rock or on leaf. Then you can design your further study on this species according to its preferences. You can represent your data in this way in visual encounter search. For example, these are the different sites. Say this is Balachara, this is Bijaypur, this is Dolu, Marua, Lakhichara, and then there is Bandarkhal. These are the sites of Barail, and I have surveyed there for, say, in Balachara area, number of survey was 17. 10 survey in the daytime and 7 survey in the night time. Okay, so because there were 2 person involved, so 17 become 34. 17 is 2 is equal to 34 hours of observation. Number of individuals of frogs I have found 165 belonging to 27 species. So that's how you represent your data, and at the end you get the overall number of species in that way. So this is the, very simple survey technique, but a lot of good results in that case. May I ask, so far I am clear, audible?
Yes, Dr. Abhijit, you were very much audible and clear.
Thank you. Thank you.
Please carry on.
So this is what you do in visual encounters. I am sure this is this is very much doable. You all can do it. You just need to make sure that you finish your survey within 1 hour time, right? So in one of those nights, while coming from our Barail Hill, in a very remote part of Barail Hill, I came across a huge chorus. Chorus means the calling frogs at one place. It's a very deep interior, muddy, slushy place with water up to my chest height, and then there are some ferns, they are coming out of the water, and frogs were sitting on those fern bank and they're calling. There are so many of them. So I caught some of them, and I have no idea what that frog is. So it took me a long time, okay, to find that this frog is exactly a new species of frog. But it took a long time to describe this frog as a new species, because I had to compare this frog that I found from Barail with all the similar looking frog of the world. That is, they are distributed in Indonesia, Malaysia, then Myanmar. Okay, so I had to collaborate with many people to describe that particular species, and that should be the approach in taxonomy. Never try to describe a species based on just one single or small observation. You need to be comparing with all the species of the world so that people recognize that you really know the science. So we described it and we named it as Micrylata ishani. Ishani means from northeast, okay? But it exactly, it's from near Borail. So I just mentioned that it's a true Sylheti frog, okay? So that's the part with taxonomy. And if you are interested in taxonomy, there is no end to your research in Northeast India. There are hundreds and hundreds of new species to be recorded. And if you want to be a taxonomist, your future is really bright. You can do a lot of research, but just make sure that what if you want to be a taxonomist, you have to be working really hard to make sure that the species that you described are really new species. Okay, so but this is about taxonomic diversity. But if you work in Northeast, you also get opportunity to know a lot about reproductive diversity, and I tell you, they are so simple. They are so simple. You know how? say for example, every night in Silchar, and if you go out of Silchar, you hear frogs calling. So it is very interesting fact that only male frogs can call. So the frog that is calling, that you see, that is a male frog. Okay. So uh, so the male frog that calls, and with the call comes the female. So female actually select the males based on their call. So that is a sexual selection strategy. Okay. So the female will select the best male based on his call, and when the female select a male frog, they will make what is called amplexus. Okay, so when you see two frogs together, you don't say that the frogs are mating, but you say that frogs are in amplexus. And these amplexus of various kinds, some frog grab the female on the neck, some frog, they will grab the female in their hip region. In some case there are lot of males and one single female. Some frogs make foam nest. Some frogs just lay eggs in the water. Some frogs lay eggs under leaf litter. So there are more than almost 20 types of reproductive behavior known from Northeast India. And if you go to the whole world, the reproductive diversity in frogs are the highest number, because they attach to both kind of habitat, that is water and land. So these are amazing information. And in Northeast India, we have some 130 species of frog. And if when I tell you this figure, out of 130 species of frog, almost 115 species of frog, we do not know how are their tadpoles, where they lay their eggs, how much time it takes to their eggs to grow. So this basic information is not available for 100, almost 115 species out of 130 species. So lot of opportunity in this field to know about them. So after they make amplexus, they lay eggs. Okay. So amphibian eggs are very, very special because they do not have any protective covering. Okay. So they are just like jelly. Okay, so that's why frogs are so much vulnerable to pollution. Okay, so because they do not have any protective cover, and after these eggs, the eggs develop into what we call as tadpoles. Now, as I told, most of the species we do not even know how their tadpole look like, and you have a lot of scope in that case. So what I want to say that this frog, right from calling to amplexus to egg laying, and uh, to egg laying, and then further up to tadpole, up to at least egg laying, all this process completes within one night. So if you are patient enough and you dedicate one night of your life to know the reproductive behavior of that frog, how much time it calls, when the male female comes, how much time they make amplexus, where they lay eggs, what kind of behavior they show while laying eggs. If you write them in a very detail, you end up writing a very good research paper within one night. Okay, so you have to be very, very careful, and obviously you can increase your sample size in that case. And then another aspect of study is tadpole. If you know that tadpole is this is the tadpole of a particular species, you can study the tadpole and know much more about it. So a lot of potential in Enuran reproductive diversity studies for you. Okay. uh, and there are also, as I told, all the male frogs call. So in India, some 430 species of frog have, you know uh, some 400 species of frog that we have, they have all different calls. So if you want to, if you are more interested in knowing technical aspect of biological science, then you can do a very good research in bioacoustics. So what you need to do, you sit very quietly in front of a frog without disturbing it, and then record the call of the frog from 1 meter distance. Okay, after you record the call, you can analyze and find out how the calls of one frog differ from another. For example, if you see this picture, these are very similar looking frog, but their call is very, very different, as you can hear just now. This is species 1. This is the species 2, Fajarvaria orissaensis. This is another species, Minervaria pieri. Okay, so you get those amplitude, the call duration, call type, so many very, very interesting thing. And with this kind of analysis, you can do just by investing 10,000 rupees by buying a good recorder. And obviously you can buy more and more better equipment later on in your study, but this is basic, right? So bioacoustic is one field. If somebody is interested to real field adventure and field research, then I give you a very interesting topic. In our Barak Valley, there is a species of lizard that is only found in Northeast India, particularly in Mizoram and Assam, that is called Tropidophorus assamensis. This is the only water skink in India. So this particular lizard, if you want to find it in the daytime, you have to walk along the very small streams which are moist, and you have to lift the rocks again and again to find this lizard. It is very difficult, but that is what we call as oticology, right? Study of ecology of a single species. And if you can do it, you definitely can do publish this result in a very big journal, because nothing is known about this lizard, whether this lizard lay egg or whether this lizard give live birth, we have no idea. So these are very interesting study. The other aspect of study is population studies. As again, you go back to your syllabus and you will find that population attributes, how what density, abundances of species. So can we actually do this? And I think you can very well do it, as we have just shown with this frog. This is a frog that live in fast flowing stream, and that's why these frogs fingers are really, really big, because they can stuck on the very slippery rock. So that's why it is called cascade frog, because that frog lives in cascades of stream. Now, this is a very special frog because that has spots on its back, and each of these spots differ from each other. So if you photograph that frog from the distance, you don't even have to touch the frog, then you can individually ID each frog. And if you individually ID each frog, then you can use mark recapture technique for identification of individual animal, and you can find out the population of the frog. Okay, and then obviously density and other things. So that's a very good study. So in our area there are so many frogs, so many lizards, so many snakes like pythons. They also have spots on their body. So if we photograph those animals, you can actually identify individual animal based on their prints, just like we identify tigers. Okay, so that's possible. We can study population, because without population data you cannot save a species. We do not know how many individuals are there, we can't do anything about it. Sometime you can actually use surrogacy approach in ecology. Okay, indirect way to answer some question. For example, I give you, there is a Chatla Haor, there is Dolu Lake. So our question may be that, is there a frog in Dolu Lake or Chatla Haor declining, or their population is good or stable? So this is the question I give you. Now what you need to go do is that every year you need to go to Dolu Lake, and evening time you have to catch frogs of a particular species, and you have to measure the body size, that is snout to vent length. That is what we measure, right? So you just measure and release it, measure, release. So first year you measure 100 frogs, second year you measure 130 frogs, third year you went there, you measure another 100 frogs, or maybe less than that, or maybe more than that. So whatever measurement you take, you segregate into male measurement, female measurement. So, and then you compare the female body size. And if you see that over these 3 years, the female body size is declining. That means their body size of female is declining. That means what? That means that female body size declining means less number of eggs. Less body size, less number of egg. Less number of egg means population decline. So you can actually use a surrogacy approach in ecological research also. And so ultimately the answer is that population might be declining. Another thing in ecology you have to remember, you cannot say everything with 100% surety. So what you have to say that might be declining. Okay, so this is what we can do. In some other research, say for example in your top in your syllabus, you will find abundance, how to know abundance of species. So for that you again may go to Barak Hill or any hilly area or Barak Reserve Forest or anywhere, and you see those area in the forest, you have very thick leaf litter. Okay, leaf litter means the fallen leaf there, and because this leaf litter holds moisture, and wherever there is moisture there are frogs or reptiles. Okay, so I want to know what is the density of frog per hectare in that area, or density of frog in that forest. So for that, what we do generally for plants, we do quadrate sampling. So in quadrate sampling, either 1 by 1 meter or 5 by 5 meter plots we make in 4 side rope, and we go inside and count the number of plants. But in case of frog or lizard, if you make four rope on 4 side and go inside that, all the frog will run away. So the frog will just be outside, so you can count them. So that may not work properly. So what we do is that we do bound quadrate. So that means the quadrate from all 4 sides, they are bound by a plastic sheet. Okay, so the frogs and lizards, they cannot go out, so you get absolute abundance in your plots. Okay, so when you do very lot of plots, many hundred plots, then you can come up with the abundance estimate of particular species in your study area. This is a fantastic research topic even for PhD also. However, we were all mostly talking about snakes. They are frogs, because frogs are very good ecological model. But when it comes to snake, then everything changes. You know why? Because snakes are nature's best kept secret. And one reason is that snakes are so unpredictable. We don't even know where we can see a snake. We can see a snake in our home, suddenly hanging on our door, or maybe outside, or maybe deep inside the forest. We have no idea where a snake will come out. Okay, so they have an extremely secretive life, and because of that, there is no standard ecological method to study snakes. Okay, if you really want to know ecology of snakes, then you have to use some very high-tech study like we are trying to do in our area, like radio telemetry. So you have to put transmitter on a snake and then follow it. Okay, that is also challenging in case of snakes. But there are a lot of opportunities also doing high level research in snakes. For example, one of the basic thing is taxonomical aspects, like what are the different species of snake occur in our area, how many species are venomous, how many species are non-venomous, is their the size of their teeth of snake are dependent on the food they eat. Okay, and you see that the number of scales on their body, so snake has particular number of scales around their body. So is that number of scales around the body related to the food they eat? What are the food of snake in an area? So because snakes are ecologically very important, because they are the best rodent controller, because they can go inside a rat hole and eat the rat. Nobody else can do it. So these are some of the things that we need to do for snakes, but still it is very difficult topic. But what are the high level research that we can do, and also some of the very simple. For example, this is a very common snake in Barak Valley. It's called pit vipers, and there are almost two pit vipers which are quite common in our area. So if you go to Baral in the night time, my highest count was in one night I have found 18 pit vipers. Okay, but that is for a long. I mean, maybe you can find less. Obviously, you are new, but over the years you will get more number of pit vipers. So these pit vipers, they will be near the streams. Okay, and there are few other snakes also. They sleep on the riparian vegetation, that means the stream side vegetation. So if you go in the night time in the forest, you just need to see those snakes where they are sleeping or where they are ambushing. So you have to take the height of the tree from how what height they are located. So if you get this data for one full year or maybe more, then you actually can find out the preference of perch height. So what kind of height this pit viper prefers to catch its prey. So this kind of research can also be done just by simply walking along the stream and seeing, and obviously not to touch it. Just do not disturb the snake. Just measure the height of the snake, and that's it. So this can be done. Obviously, some other research. For example, this is the most uh, enigmatic and the most majestic snake of the world, king cobra. So we are studying the ecology of king cobra in Uttarakhand, and we have found very interesting results. For example, in Northeast India, king cobra is the only snake. I mean, king cobra is the only snake that makes a nest, and this is in Northeast India. King cobra uses bamboo leaf to make nest. In Uttarakhand, we have found king cobra in some area they are using oak leaf, and in some area pine needles. As you see left corner, pine, they are using nest, making nest which is as big as 5 feet wide and 3 feet high. The large, very large nest, they are making with pine needles, and in Assam or in Mizoram they are making it with bamboo leaves. In our area also king cobra occur, but we do not know where these nests are located. So that if somebody really want to do a challenge and very very strong field oriented guy or a girl want to conduct a research, so you, if you find a king cobra nest, you can go start monitoring them, aware people not to kill the snake. It's a lot of work you can do in that way. Okay, but this is really really great work to save a species. Just imagine a snake without hand or a limb, it makes such a large nest and stay with the nest until their babies comes out. Okay, so that's a great behavior. The best question in king cobra is that why king cobra make a nest? That is the question. We nobody can answer this. Why, being a reptile and a snake without limbs, why it makes a nest? It could have gone in a tree hole and lay the eggs. Why it makes nest? So dangerous, so much work, why it do that? If you answer this, you publish a nature paper. So there are some emerging threat. Okay, emerging threats are coming up every day. Now lot of frogs that are found that are deformed. Okay, so if you want to take a simple research topic, you can just go in around in your area, say Pachgram paper mill area, Dolu tea estate, many other area where pesticide is used. Okay, in agricultural field, you can go in the night, find out frogs and check if they have any deformity. If they have any deformity, there are monographs available to identify what kind of deformity these frogs are having. Okay, so that's how you can do another kind of study, that knowing deformity and predicting why it is happening. So that these are new emerging field in research. Okay, and that's probably my last slide, is about 5 research topic today I want to give you, and these are just 5. You can make 50 out of many, 50 after that, but these are mostly related to your maybe your syllabus also, or what I have told you so far. Okay, so maybe one research topic is Just simple oviposition sites in 5 perianthropic frog species. What is perianthropic means? Perianthropic means the frogs that lives near human habitation in village area or in a slightly disturbed area, like toad, some water frog, some common frog, some tree frog, which live in and around our village or cities. So we do not even know where they lay eggs. So you have to find out just the list, this place where they lay eggs. So that is called oviposition sites. And you can do if this is 5 species, you can do it for 50, for 100 species. Okay. Then you know that Mahasharak is coming up. So there will be road going along Boreal Reserve Forest, and similar kind of road is everywhere in Northeast India and in Barak Valley also. So if anybody wants to study roadkill mortality, because you all know that snakes are one of the most important for that. The reason is that snakes animals that takes heat by body surface. So snakes comes on a hot surface of the road and take the heat from the hot surface. That's why they are called as thigmotherms. Okay. So these animals they come on the road and the black tar road and sit on the road, and then the vehicle comes and kill them. So if anybody wants to do a work, that just take a GPS and walk along the road and just record the GPS location of wherever you are seeing any snake, any frog, any lizard, or any other animal dying on the road. You just record the GPS location. After you do it for 6 months, you will see a pattern. You will see that at some place there are more roadkill, some place there are less roadkill. So you can then make a map of that area showing that this is a this is a most affected area, this is less affected area, and you can show that in the map and produce that map with the forest department. And you please tell your DFO there that, see, this is the area where most animals are dying, please do something. Okay. So then the third topic is simple tadpole density and habitat selection. Now, every year in June, July, if you go to any pond, any stream, any water body in Silchar and Barak Valley area, you will see tadpoles there. That means frog larva. Okay. Now, can you do a study on density? Can you try to find out density of the tadpoles? So, what you need to do, you may need to make 1 meter, 1 by 1 meter, a kind of a net or mesh. You just can pull with each stroke. You pull up the tadpoles, now count the number of them and release them. And you do it at multiple places. You actually can find out the density of the tadpoles and what kind of habitat that tadpoles are selecting. Some tadpoles live in fast flowing section of stream. Some tadpoles live in pool section of the stream. So all different habitat types in the streams that tadpoles can use. So you can very nicely do it. Obviously, I will obviously give my favorite topic, breeding biology of Macrylata ishani, because as I told you, at the time when I collected, I could not record the call of the frog. I could not see the where this frog lay eggs. I could not see where this how the tadpole of Macrylata looks like. So it's all left to you now. You do it, and not only Macrylata ishani, you can do it with 100 species of frog in Northeast India. Okay, studying their breeding biology. Then another study is amphibian deformity in tea gardens and agricultural fields. So you can select your different study site, go to tea garden, go to agricultural field, study them and find out if there is any deformity. And if somebody more want to do some very unique research, for example, you have a very small tiny frog found in Assam, Darang district in Orang Tiger Reserve, that is called Orang sticky frog. That frog secretes a very sticky substance, which if you touch, it will be there in your hand for 3 days. Okay, with even with soap, you cannot remove it. Such a sticky material. In Southeast Asia, people are trying to make bone glue for broken bone with the frog sticky substance. It is so much stickiness. sticky. So this kind of biological research can also be done from herpetology. Now, I probably left all to you, and probably you all know that the only medicine for snake bite is a snake venom. Okay, so snake venom is another very important research topic, and it can be done. There are a lot of snake that mimics dangerous species, like wolf snake mimic trait. So that's called Batesian mimicry, that you can study in your area, and these snakes are available in your area. And recently we have rediscovered a species after 129 years from Assam, Arunachal border, that is Assam killback. Okay, so nothing is known about this species, no ecological information. Can anybody take up this research that to find more of Assam killback and map the distribution of the species? That will be a great contribution to the species conservation. So these are some of these topics you can choose, and there are lot many, and you are always welcome to discuss with me. Come to me, send email, you can ask me question even in Sileti also. So you can do a lot of things. You have to open up to do research in wildlife. Wildlife is a branch of science where anybody can work. It's not the zoology student, it's everywhere. Botanist, biotechnologist. I heard that GC College is one of the premier institution where you have lot of, even you have molecular tech tools and stuff to do, right? So why can't you can do your research right there in your GC College. You can do molecular taxonomy at your lab, you can do PCR, you can identify new species, you can generate, and you can be center of attraction for everyone. And I really want that my alma mater college should be the best in Assam. So thank you very much, and if you have time and energy, you can ask me some question. Thank you.
Thank you, Abhijit. That was really wonderful. We have time for a question answer session. So please close the screen sharing option. You have closed it so that we will be visible to you. Are we visible to you,
Is it
Doctor?
closed now?
Yes,
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 yes.
yes.
That's okay.
Can you see us?
Yes, now I can see. Yeah.
Okay, that's great. Can you see the comments also? On screen comments, can you see?
Uh, sound,
Sound.
your sound.
Hello? Am I audible to you?
Yes, yes, yes.
No.
Yes, ma'am.
Dr. Abhijit, am I audible to you? He is not
No, I
listening.
can't hear.
You can remove your headphone. Dr. Das, you can remove your headphones. I think he is having some connectivity problem. So I would now request Aradhana and Vishwa, once Dr. Das is back with us, to coordinate the question and answer session, because we have got a lot of questions on the screen. So I'm sure you might have noted down all the questions. So you coordinate the question and answer session with Dr. Das. He will be right back, I think.
Okay, ma'am.
Madam, am I audible? Prema Madam?
Yes, yes, yes,
Uh,
yes.
can you please call Madam Dr. Das and tell him to reconnect again?
Okay, wait, I'm
Just
calling.
call him.
Hello,
He's
madam.
back, I think. Oh, so you are back. Yeah, there was
Yes,
some connection problem.
Can you hear me now?
Yes, yes, you
yes.
are
It's
audible.
okay.
You are audible.
Perfect.
Okay.
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 Okay. So, Dr.
Thank you.
Das, now I would request two of my students to coordinate the question and answer session with you. A lot of questions are coming for you.
Yeah.
So, Vishwa and Aradhana, please go ahead.
Very good evening, sir. That was
Yeah.
really a very exhilarating session and
Yeah.
got to know so many new things. Sir, my first question to you is before that, you talked about Mecphileta aishani. Is it the same species? I guess I have read in a famous newspaper a few months back that a university collaboration was done from US, I guess, and then this species was discovered, sir.
Yes.
Is this the same
Yes.
species?
I think so. Yes, because there will be only one Microleta Ishani. So
Yes, sir.
yeah.
Few months back after
So
Corona strike, I read this somewhere, sir. So, you talked about the segregation process of male and female frogs. So, sir, can you just
yeah.
put some light how to segregate the male and female species,
Uh,
male and female
that's
individual?
Yeah, that's a good question. So, in case of amphibians and also in most case of reptiles, we do sampling mostly in the breeding season. Okay. So, breeding season, mostly the females will be carrying their eggs. So, their belly will be bloated. Okay. So the belly will be full of eggs, and we use a very small torch light, very less powerful torch, almost like a pen size. So we switch on the light, and we see through the skin. So if there is a egg inside, we can clearly see the eggs mass inside the body. So it's a very easy technique for frogs to identify male and female. And another very interesting or easy way is that any frog that is calling, that is a male frog, because females they cannot call. Yeah.
Okay, thank you, sir. This my next question to you is when it comes to frog, you said that we can measure it by like we can put some plastic cardboard there so that it doesn't move while we are measuring the abundance. So what about there is a species of frog which jumps very high. So in that case, what we can use?
Well, see, when we this slide was particularly for we discussed about leaf litter species. Okay, so our question is leaf litter herpetofauna. So the animals that live within leaf litter, and the frog that you said very nicely jumped, that is mostly a tree frog. Okay, so tree frog quadrate is a not good method to sample for tree frog. That should be a different method. Okay, but the frogs that live only under leaf, there are some snakes like coral snake, they live under leaf. So these are the leaf litter species, and abundance of leaf litter animals is very very important because their abundance is related to moisture, and moisture is related to the growth of the forest. Okay, so
Yes.
you can have a surrogacy approach using frog as a model organism to know moisture in a forest habitat. Okay, so that's what I said, like, it's only about abundance of leaf litter herpetofauna.
Sir. So you have shown us some tools which we can use while we are in the field. So I could relate that to what extent we could use those tools. So sir, I just want to know how vernier caliper can be used in the
Oh,
field.
okay. So vernier caliper is can be used in the laboratory as well as in the field. So as I told you, in the field, if you have want to measure the snout to vent length, okay, so you can just use this vernier caliper to measure the length of the frog, and then you release the frog back in the forest. So you need not to bring the forest in your laboratory. Okay, so for ecological studies, you can use vernier caliper for frog measurement in the forest.
Okay, thank you, sir. So about the Barai Wildlife Sanctuary, I personally went to Barai with Nilendu, sir, in my first semester, so I could relate to the images you have shown to us. And thanks to Nilendu, sir,
Yeah.
he gave us this opportunity. Sir, my question to you is, while we were there with our seniors in my first semester, we went to a
Hmm.
forest guard office. We saw, I think, if I'm not wrong, there was just one or two guards over there. So my question is, how, what do you think, like the legislation is failing to protect, since it is a huge area, and thus there were just two officers controlling the whole area. So do you think it is hampering the conservation process? Lack of
Yes.
mass?
Yeah, I mean, I understand this, what you were saying. Uh, it's a very difficult question, and I don't have an answer for this. But one thing that I feel is that it depends on priority. Okay. For example, we as a human being, we are more biased towards large animals. Okay. So if there
Yes.
is rhino, if there is tiger, if there is elephant, there will be more protection in a particular area. But if there is frogs and snakes and small birds, there is hardly any protection there. So Bareilly is one area where earlier there were tiger, there were elephants, but slowly we lost them. Okay, so we don't have any tiger or elephant now. But maybe if we protect them better, they will come back one day. But as of now, we don't have. So priority is least in this kind of area for protection.
Thank you.
But I feel that once more and more research done, more and more findings are done, more people are aware, like you are aware now. So forest department is bound to act to stop all this kind of illegal activity.
So, my next question to you is, this is my personal question. What I feel that the research that we undertake, or any students undertake, it is basically to get a PhD degree. So, what I'm trying to say is the research is mostly based on academics. Post academic, this
Hmm.
research methodology, and it is not being used for any conservative purpose, for any other purpose after post academic research. So you think that this is some of the factor that the academy is, the research is not that popular, because mostly they do this to get the degree only
Yeah,
for the academic.
as I told in the beginning, probably that research has different dimensions, and there are if you see, there are more students interested in biotechnology, microbiology, molecular biology. There are more and more people are interested more than much more than ecology. Okay, so the reason maybe is just because that we cannot relate ecology with our daily life. So that's why what we do, we cannot keep doing it for whole of our life. So that's why we may get least interested. But as newcomers are coming, they are very bright students nowadays. So if they, from the very beginning, we start teaching them from the field, we tell them the story from the field, start allow them to do small experiments, experimental studies. So experimental studies make ecology as interesting as molecular biology. Okay, so then people will be more interested. Then people will never leave the subject and go to another thing. And they will publish more. And once you publish more, you need not to go anywhere. You will get everything what you do. So these are
Yes.
all related, and they starts from beginning. That is, BSc is a very important time when the students need to start.
So my next question to you is about your experience when you started initially this whole process. So what sort of difficulties you faced, since you belong from Barak Valley, so you know that much popularity is not there. So did you feel like, regarding any tool or guidance, what sort of difficulties you faced, and if you faced, how did you overcome those?
Well, yeah, I mean, I got. So, what I feel is that yes, I am from Barak Valley and I am from a village. There are difficulties for me there were difficult time to, when I thought about my good friend and one of my best friend, Dr. Amitabh Bhattacharya. He selected microbiology and I selected wildlife. So it was a very difficult for me to select wildlife for an MSc again, and so but then that was my interest, and I wanted to do my interest, and my family was very supportive to that. So that is one prerequisite, I think. And the next thing is that uh, I always try to make my weaknesses as the best point, because the Baral Hill range from where I started, nothing was known, and I projected that in this way only. I wrote to people that nothing is known, so support me. So I will go to forest and I will bring the information. So I think this is our opportunity. Our weakness should be our strength. So still now there are so many small patches: Barak Reserve Forest, Inner Line Reserve Forest, Katakal Reserve Forest, and many other places. Okay, so nothing is known. Okay, and I am sure if you guys are interested, so you can make those as your strongest point. You know, but you
Mhm.
need a very you need a dedicated mind.
Okay, I will interrupt a little bit. Vishu and Aradhana, please take up the questions on the screen. A lot of questions are coming. Please
Okay, ma'am.
take up those questions. Yeah, we're running out of time.
Now, what to do?
what what are
Yes,
avoid
ma'am,
Hello.
ma'am, I have a question to ask to our respected speaker, sir, if you would allow me to ask.
Yeah. Of course, please ask and then
Okay.
we will finish.
Good evening, respected speaker, sir, and to all my respected professors present here. So my
Yeah.
question is, sir, if you could please tell me something about the snake bite protocol so that it can help the common people and spread awareness.
Okay, so this is a whole dimension. I did not talk much about it, but yeah, snake bite is a really big challenge, and that is one. Snake is like Amresh Puri of biological research. The whole game changes due to snake. Okay, everybody would have loved frog and lizard and birds and everything, but nobody want to go to forest because there are snakes. Nobody want to study herpetology because there is snake. Okay, so that's a bad thing. But truly speaking, only very few snakes are venomous, and out of these very few venomous species, only few live in our human habitat. So if in Barak Valley, if I talk about, there is cobra, there is a crate, and there is pit viper. So 3 to 4 species are venomous, and they are in the village areas of our Barak Valley. So if you can only identify a cobra and a viper and differentiate them, mostly you can help people tell the story that these are the two venomous snake and rest all are non-venomous. So that's our job as a responsible citizen. We can spread this message. And the other thing is that snake bite. In India, something like 50,000 people die from the snake bite every year, which is a huge number. You see how much hue and cry due to corona death, even for one, but 50,000 every year, people are dying. Nobody talks about it. So one of the reason is that our lack of awareness about snake bite. Okay, because when people get bitten, people prefer to go to Oja and Baba, and not to go to hospitals. So that's a problem, because now in government of India had made free supply of antivenom in our medical colleges, and that is also now coming to our hospitals. So people should, if there is a snake bite, people should immediately go to hospital, not to waste any time doing any business, because in snake bite the most important thing is time. Okay, and if you go to Baba, call Oja, then you are losing that first golden hour. Okay, and in that, if you reach to hospital within that first 1 hour, then mostly the patient will survive, but that is not happening, and that's probably the reason that people are dying. And also wrong treatment, like people cut the wound, people tie the hand or leg very strong, I mean tourniquet. So all these things should be avoided. And if we do that, our snake bite case will reduce a lot. So it is only a matter of awareness, I feel. And I think young students should take up this challenge of making people aware about snake bite. So the best treatment, best what I call best first aid for a snake bite is a mobile phone and a car or a bike key. So your job is to call doctor and go there. That's it, and rest all doctor will do.
Thank you, sir.
So many students
Okay,
are
sir.
asking question. You can take up
Yes,
a question from the screen.
Oh
yes,
yeah.
ma'am. Yes, ma'am. I'm
Please,
reading.
please take up questions from the screen. Someone is asking, sir, what abiotic and biotic factors influence behavioral and physiological changes in the reptilian species of Barak Valley?
Okay,
I,
that's I think that's a two PhD. Okay, but this is a very important question. What are the biotic and abiotic factors? So it may be reptile, it may be birds, it may be frogs, whatever species it is. But when you go to forest, you remember what are the factors that you meet. So, for example, if you talk about abiotic factor, so these are temperature, these are humidity, these are rainfall. So these are the abiotic factors that you face. So for example, if a frog calls, okay, so frog is calling and you are recording the call. So if there is the air is very dry, there is less moisture in the air, the frog will call very less, because frog knows that today is not a good time to breed, because today there is not enough moisture, it will not rain. So it will call very low pitch. And if you come back from the forest and you say that, oh, I have recorded the call, but that is not exactly the call. But after some time, when the rain comes, then the frog will go mad, it will start calling like crazy. So its call will be much more different than call at a less moisture. So you have to take multiple sample in any ecological experiment, not one sample. Okay, so that's what is abiotic factors. And biotic factors, like what are the predators available in an area. Okay, for example, if we go to a river or a stream, we get frogs there. Okay, now why a frog lives in one particular area where it lives? For example, we live in our house. That means we have all our things in a house. We have all the facilities in a house. So frog is living in a stream means it has all these facilities, right? So how can you know that? So what you know, you can catch a frog, and there is a gastric pipe method. You can put a pipe inside the mouth of the frog and put water, so the frog will vomit. So with that vomit, the gut content of the frog will come out. Okay, so then you take the gut content of the frog and you see what are the food that frog is eating. Okay, so you find that frog is eating beetles, the frog is eating some flies, frog is eating this. So you conclude that the frog that you have found is a terrestrial species. But if you find a frog eating the insects which are found in water, that means that frog is a truly aquatic frog. So that's how you do. These are biotic factors. Okay, so that's how you do your experiment. Although it's a very small experiment, but still it gives a very important information about biotic and abiotic factor influencing distribution and abundance of species.
So next we have one more question from our viewers. Could you please throw some light in identifying reptilian species with respect in using tools of molecular taxonomy?
Oh, okay. It's a very good question, and I hope Debjani will be.
Devdasanda.
Yeah. So molecular taxonomy is now everything, So now if you want to publish in a good journal, so they will ask for DNA data. Okay. So it is very important, crucial. For example, Snake has a very long body and body covered by scales. So we have we count the scales to identify things. But okay, so over these days, what happened is that this kind of identification is got saturated. Now we need more and more data. So that's why molecular taxonomy is one tool. So you have to collect tissue or a small scale. You have to do the PCR. You have to make the tree and see the phylogeny, and you can identify the species, how much different it from the closely related species. So that is a whole new branch of science that is unfolding in front of us, and that's why more and more species are now described recently. Hi, Pratik.
Thank you so much, sir.
One more question is coming up.
What
Aradhana,
com-
go ahead.
Yes,
Oh,
sir.
so,
There is a question from Shantani. She is asking, what is the composition of the substance secreted by the orang sticky frog?
okay. So if you are interested in biochemical research, that's one topic for you. Nobody knows what is the substance secreted by orang sticky frog so far. But one thing is that, the research, it's the same frog, that same sticky frog which is found in Thailand. So researchers have extracted the sticky substance. Now, this has a lot of implication in osteology, because for the doctors in osteology, the main problem they find is that there is no glue available in the world which can help us stick our bone in aqueous medium. So our inside our body is an aqueous medium. There is lot of water there. So there is no glue available which uh, can be used to stuck our bones inside our body. We cannot take our bone outside and stuck and go inside. No, we have to stick it there inside if there is a bone break there. But frog body is always moist. It is also an aqueous medium, and they secrete such a sticky substance. So that is very unique thing. Now, then if we can now extract this particular biological compound, then this glue we can use inside our body to stick our broken bones. So that's the science that lot of, even NASA is doing research on this. There's a project going on.
One question is coming up from Dr. Subhash Devnath. He is a faculty member of chemistry department. He's asking, majority of snakes in this region are non-poisonous. Pit viper's mouth looks black in color, and people in village believe these are very poisonous. Just want to know whether these are poisonous. Pit
Okay.
vipers.
Okay. So, sir, that is a very interesting thing. Your question has revealed another interesting aspect. So, the snake that you are talking about, you must have seen a green snake, and which has a black mouth inside. So mostly pit vipers are green in color. So we probably, that is the conclusion that it is pit viper. But the green snake with a black mouth is actually not a pit viper, but it is called green cat snake. Okay, green cat snake has a green body and its mouth is very dark color. Now, cat snake is not venomous, but pit viper is venomous, as you said. Now, so if you show your black coloration inside your mouth, so obviously it looks like I am very dangerous or poisonous. See, my mouth is black, so I am dangerous. So yeah, I mean, it's a threat display. It is not related to anything called venom or poison, but it is a threat display. Many snake that makes a hood like a cobra, but they are not venomous. So these are called, kind of Batesian mimicry, or other kind of mimicry, where non-venomous snake mimics a venomous snake. In this case, green rat snake mimics like black mamba. Black mamba also has a mouth which is black in color. So that's the answer. I think the snake that you have seen is a green rat snake.
Okay,
What?
So
Bharti
we have this,
Chakravarti.
one more question we
One
have.
more question. Yes, yes. You just
Can you, how can we easily, is there an easy process to identify the poisonous or differentiate between the poisonous and non-poisonous snakes?
Okay, that's a question every time, all the time. But obviously, there is not a easy way to identify venomous and non-venomous snake. The easiest way is knowing the venomous snakes. So as I told, in our area, there are only two group of snake which are venomous. That is, one is cobra group and one is viper group. Viper group snakes are very easy because they have a triangular head and body is very short. Cobra group of animals are little difficult, but crates are the snakes that only comes out in the night time. So if you are in walking in the evening time or night time, please take a torch and walk, because that is the time for a crate. So crates are shiny black in color. So any snake you see in the night time that is black in color can be dangerous. So these are very simple thing, and one very simple key I will give for all the snake of Barak Valley. Any snake that you see in Barak Valley that has long stripes on the body, stripe means very long line. So any
Yes.
snake that has a long stripe on the body are all non-venomous. So that's the simple rule for identification of a non-venomous snake. Any snake with a long stripe on the top of the body, all are non-venomous snakes in our area.
I have one question,
Yes,
Vijay.
yes.
Can we identify a poisonous snake from observing its tail region?
Hmm.
Tail
Okay.
region. From the
Yeah.
tail
So
region.
I remember that in old this our textbooks and all, earlier we
Yes, yes,
used to have identification of poisonous and
yes,
non-poisonous
yes,
snake, but
yes.
those only in sea snake we can identify by looking at the tail, because sea snake has a flat tail. So flat
Okay.
tail is because to swim in the water, so they have a flat tail. So and most of the sea snake, most of them are venomous or dangerous. So that's why only tail is helping in identification of sea snake. But all other snake that lives in our land, that is terrestrial snake, we cannot identify by looking at the tail. But tail is important in other way. That is for secondary sexual dimorphism. Tail is important.
Okay.
For
Okay.
example, in male snake has a long tail, but female snake has a shorter tail. So that's a dimorphic character.
Okay.
Yeah.
So thank you. Any other question?
Yes, ma'am.
Vijay
I
Brahma.
do have one.
Okay. Professor Brahma has come up. He's asking, Professor
Yes.
Brahma is our colleague, Abhijit, Dr. Abhijit. Professor Vijay
Yes.
Brahma is one of
Yes.
our colleagues. He's asking some reptiles show repeats sensing,
Senses.
sense,
Senescence. Like,
okay,
Like
like
skink.
skink, whereas some reptiles show very slow sensing like turtle. Why is that so?
Reptiles show some rapid sense. Huh?
Rapid
Sense
sensing like skink.
skink. And whereas
Still, it's
some
very
reptiles.
difficult question.
No, no. What I am not able to understand this.
Yes,
Um,
yes.
is it about growth or movement? Sense.
No
Sir, what
idea.
I
I
can
don't
make
have
of
it.
this question is
Sense.
about the movement, I think. So it's like
Oh, is it? Okay,
movement.
okay. Movement. Okay, okay.
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 Rapid
So
movement. Rapid
yeah, yeah.
movement.
Yeah, it's correct. Actually, there are some reptiles are very fast and some are very slow. It's probably related to the way of lifestyle, because uh, turtles and tortoises, if you see their body shape, their bodies are like very very well protected. Okay, so probably turtles and tortoises are the best protected animal in the world. Like, they have a very, very strong shell Even we have seen that even leopards cannot break the shell of a tortoise by biting it. It is so hard. Okay, but only animal that can tortoises cannot survive is human being. Okay, and especially I think Bengalis. So but in that case they have a very very strong shell. So they whenever there is a threat, so they take their head and feet everything inside the shell, and they protect themselves. Okay, but in case of skink, okay, so skink has to run away, because otherwise they will be eaten by any other things, because reptiles in the forest ecosystem, they are both predator as well as prey. So they have to run away, otherwise there are a lot of snakes or birds that eat skinks. So that's why they are really fast to go. But a tortoise need not to run, because if anything comes to eat it, that will immediately hide himself inside their own shell. So they are better protected. So it is not necessary to run away, because you already have a protection.
Yes. So
Pratik.
next we have a question from Pratik. He's our senior. That at what elevation can we expect king cobra that makes nest with oak leaves and pine needles in Uttarakhand?
Okay, so that's a very interesting thing. So recently, the last week only, there is a report of king cobra from 2,500 meter. So this is now 2,500 meter was the highest elevational record of king cobra. The lowest elevation is as low as 20 to 30 meters, and highest is 2,500 meter near Nainital, probably and in Northeast India, I have recorded king cobra up to 2,000, 1,900 meters from Nagaland. One or two.
Any more question?
I have one last question, sir. As we know that in India, research is not that much popularized. I guess only 1% of the students go for research work, if I'm not wrong. So now we are hitting, like we are being hit with COVID-19. So more grants will be reduced, grants will be shrink with the government because they have some other expenditures to do. So how you see that, what will be the impact of this in the research work, which is already very fragile?
What you wanted to say, that with COVID pandemic, research will be reduced?
Yes, sir. Research will be reduced
Yeah,
because of
yeah.
the
So
shortage
that,
of fund and all.
yeah, that's true, because funding is shrinking.
Mhm.
Funding is shrinking, and there may be, because obviously it impacts our economy. But what I want to say, that if you have a real mind in research and you really want to do something, you can do small experiments near your home. And today's talk is all about our young students, who hardly get funding, because they are small, they are kids. So the way they start is from their backyard. And if you read Darwin's book, the Darwin, our father of evolution,
Yes,
he
absolutely.
used to do a lot of experiments near his house. Okay, and I am fortunate enough to go to his house and see his house. So what he used to do, he used to encircle some area, and he keeps some area open in his backyard. So he encircled the area that is completely devoid of any predator. There is no cows coming, no cattle coming, no birds coming. And then he used to see how many plants growing in an encircled area and how many plants are growing in an open area. Then he used to compare. So this kind of experiment and this kind of ideas do not need funding. This kind of small research only needs you to be driven, from core of your. I just want to quickly answer one question by our respected sir Saibal. He is a great person, and I really appreciate him, because he was a college teacher, and he made so much contribution in herpetology of our country. He mentioned that Pope pit viper is available in Baral. Yes, I am fortunate enough to find Pope pit viper from Baral, but that was only one record. But so far, most of the record of pit viper from Baral is spot tail pit viper. But there was only one record of Pope pit viper from Baral by me.
We are so fortunate that Sir is here with us. He's listening to us, I think, I hope. So thank you so much, sir.
Yeah.
Dr. Shoibal Gupta, sir, thank you so much. Next, any more question or otherwise we will wind up our program. So, Vaishakhi, what she is asking, sir, you said that a particular frog can call differently in different conditions. Is there a way we can conclude that it's the same frog calling differently and not the another frog's call?
Okay, it's I just maybe this is last question. So yeah,
Yes.
this is a very good question. So see, if a frog calls, it is as I said, yes, that will call at a different condition. So when you are recording the call of a frog, you also need to record the atmospheric temperature. What is the temperature at which you have recorded the call? Okay, so because with the increase in temperature or decrease in temperature, the call pattern will vary, but the call type will not be the same. Okay, for example, when you are feverish, you talk in a different way. When you are very energetic, you talk in a different way. So it's just like that, but otherwise your voice will remain the same. So frog is also like that. Species specific differences remain the same, but it's only the pattern of the call that will differ. So you have to know at what condition you have recorded the call, and that's why multiple observations are important.
Thank you so
Thank
much, Dr. Das. Thank you, Vishwa. Thank you, Aradhana. And we
Thank
have
you.
come to the end of our webinar today. So now I would request our senior faculty member, my teacher also, Dr. Nilendu Dhar sir, to please extend vote of thanks. Please, sir.
No need.
Everyone is laughing.
thank you. Hopefully, I am audible.
Yes, yes, sir.
Yes.
You are.
Yes, sir.
Um, Firstly, it is in some sense a nostalgic moment to see Obijit again, and his second presentation I am attending. The first one also with students that in WII, hopefully in 2015, I think. And at that time, I'm sitting on the back, and Abhijit has taken the first session. And after that, he was there along with us the whole day and coordinating the whole day program with us. And he's taking to the canteen, sponsoring the entire session. And I remember all these things. That was one thing. Another moment I could recollect, because that's why I'm feeling nostalgic. I recollect that during the BSc days, hopefully, Abhijit, you can recollect, we
Yes,
have
sir.
visited Jatinga.
Mm.
And
My
at
first
that
trip to
time,
Goa.
yeah, and that time we used to ask a lot of questions. Enthusiasm was same that time, and now being a scientist D, you are the same person. And that is the thing. The enthusiasm lies there since the beginning, and you are maintaining same enthusiasm here in Lee at that level also. And that is the thing I feel which will be the main boosting factor in your lecture. While I'm attending, you are making the research so simple and easy, starting from the very basic and making the research easily approachable by a budding scientist, budding researcher. So you told me not to give you thanks, so I am not thanking you, but it is in some way generate energy to us, even a teacher, anybody who is watching you will get some energy, and that is the main thing which will inspire everybody to carry on with research. How is Papia?
Yeah, Papia is fine. So that's what I mean, I'm so much indebted to GC College in that way because I got my wife also from GC College. So
Yeah,
extra energy.
that is the thing. She's there in Dehradun.
Yes, sir. Yes. Lockdown.
Okay, okay. So that's I'm not.
Right.
It is a long session. I'm not prolonging it anymore. I thank the two student coordinators here who are very nicely coordinated the session. I thank Devprita for nicely arranging the session. The HOT deserve thanks. The principal deserve thanks, and mainly the key person who is on the background, Dr. Devashish Sharma, and his team, Nirupam, who are organizing all these webinars. They deserve a great applause. So, with all these words, Abhijit, thank you so much,
Thank
and
you, sir.
be in more sessions, simultaneous session. Hey, Devashi is here.
Yeah. Thank you. Thank you, sir. I'm good.
Devashish is very popular for the wrong reason these days because he has become a COVID patient.
Yeah, yeah.
For this reason, he was popular for giving interview for a lot of
Yeah,
organized
yeah, sharing my experience.
experience. Thank you, Devashish. Uh, so, with these words, I stop here. Thank you so much.
Thank you, sir. Thank you.
All these participants,
Thank you.
okay. All the
Thank
participants, I
you.
am thank
Thank
all
you.
the participants in these sessions. Okay.
Okay, thank you so much,
Thank
sir.
you.
So, thank you, Dr. Das, once again. I hope to meet you live,
Yes.
not virtually, someday
Yes.
in the college, in the
Yes.
department itself.
Yes.
Okay, so
Thank you.
thank you all. Thank you, madam. Thank you, sir. Thank you all. So we
Okay,
conclude our
madam.
webinar here.
Last but not the least, last but
Yes.
not the least, Abhijit, please do visit our college if you get time. Whenever you come
Sure.
to your home, then please visit our college. You inform us and please visit. Okay.
Yes.
Thank you so much.
Thank you, ma'am. Thank
Thank
you.
you.
Bye, ma'am.
Bye. Bye.